Good evening, welcome to tuition2u.com. You are now with V. Sundram, and in this video we are going to discuss about SPM 2022 and Biology paper 3. So Biology paper 3 is something new. It is in the syllabus all the while, but previously it was fully written exam. Your batch is the first batch who's going to sit for actual practical. That means you must do the practical. Based on the results you obtain, you need to answer the rest of the question. So because this is the first batch, so there's a lot of uncertainties, a lot of confusion going around. So I've decided to do this video so that you have a general idea of how to answer the paper 3 question. So let's quickly dive into the question. So this question that I'm going to discuss, I assume most you can download it later from my page, or I assume most of you already have the question with you. So this is if you go to many social media forum, SPM forum, many teachers, many students are expecting experiment related to DCPIP, ascorbic acid, vitamin C question to come out this year. So no one knows what is the question until the day the students sit for the exam. So don't believe in all of this Solan Bocho and onwards. But they looks like there are some experiment that more people are talking about it. So anyway, the good news about paper 3 is there is a certain techniques you can use for every experiment. So even if you don't know the experiment, at least last time, like the practical, the paper 3 last time, they don't have any material, any apparatus between them. So they have to, if they don't know the experiment, they don't know. But as for you, you have the material in front of you, you have the apparatus in front of you. At least it's easier for you to predict how to conduct the experiment. If they give you, let's say, a fruit juice, apple juice and orange juice, or orange juice, pineapple juice, that you automatically know that is the manipulated variable. It's in front of you. A different story for last time, because if they don't know, they don't know. There's nothing else they can refer to. You have the material apparatus in front of you. Generally, some of the students are very concerned. What if it's unguided experiment? It's in front of you. Use it as the answer. So generally, you should treat paper 3 as a bonus paper, as my personal opinion, bonus paper. Just follow the instruction that is given, do the experiment accordingly, and use the techniques that your school teacher explained, that I'm going to explain, and get full marks, or at least almost the full marks. It's supposed to be an easy paper. Okay, so I'm explaining in terms of biology perspective. Other subjects I don't have any comment. For bio, paper 3 you can get the full marks. So we are going to discuss this question, which is DCPIP question that you can download from internet, or most probably you already have it, or you can also download from my page later. So let's quickly move into the question. So the first thing is, they give you the first page, you have this candidate checklist. So they have a list of materials and also apparatus. Just make sure you have all of these materials in front of you. At the same time, if there is some electronical devices, let's say stopwatch for example, make sure the stopwatch is working. Just check whether the electronical device is functioning properly or not. We want to make sure when we conduct the experiment, everything goes smoothly. The teachers should have already checked all the materials and apparatus, but you can do your own check. Better safe than sorry. So you can make your own check within the time that is given. So make sure you have all of this. Then once you already have all of this, you can start to plan the experiment. We don't know whether it's going to be a guided or unguided experiment, but the difference between these two, guided and unguided, is basically the first question. If it's unguided, you need to inform the examiner how do you control manipulated responding and control variable. That's the only difference. Other than that, every question remains the same. So we'll tell you the techniques how to get all of that. So if you go to the next page, So this is an experiment related to DCPIP. So when you see DCPIP solution, you should automatically know this is something to do with ascorbic acid, vitamin C. Always read the question. Many students make a mistake. They rush through the question. They go straight to this question here. The question doesn't begin here. The question begins right on top. Read every sentence because many times the first paragraph gives you a lot of clues. So if you read the first paragraph here, you have to carry out an experiment. You have to carry out. That's instead of instructing please carry out, you have to carry out. Okay, so you have to carry out an experiment to determine. So the moment you see the word to determine, to investigate, to experiment, that is the beginning of the main explanation. So to determine the contents of vitamin C. So to determine the contents of vitamin C in fruit juice. So the first paragraph here is very important because it tells you what is the whole experiment about. You are supposed to determine the contents of vitamin C in fruit juice. So the sentences that I've underlined actually is giving me information that these contents of vitamin C is the contents of vitamin C is the responding variable, whereas the fruit juice is supposed to be the manipulated variable. So we are going to use different types of fruit juice, that is manipulated variable. The contents of vitamin C is the responding variable. After you get that two information from the first paragraph, you look at the diagram. Normally from the diagram you can get the constant variable. Sometimes, for example, here they might have written 1 ml of DCPIP solution. So that 1 ml is supposed to be the constant variable. But in this question, the diagram doesn't have that information. But normally, from the diagram, you can get the constant variable. In your actual question, check the diagram in case they have some clue for constant variable. So far, we know what is manipulated. We know what is responding variable. Let's continue with the procedure. The procedure is important because, yes, you need to follow the procedure to get your experiment result. But at the same time, in this procedure, there is a lot of information that you need to take out. What is the manipulative variable, responding variable, constant variable? What is the precautionary measure? These four information, you can get it from the procedure. So read the steps one by one, and in case you identify any one of these variables or any one of these precautionary measures, you can underline. You need it for some of the answers later. So let's start with the first sentence. Put 1 ml of 1% DCPIP solution into a specimen tube. So when I read this first sentence, I automatically understand that 1 ml of DCPIP solution is the constant variable. So this is supposed to be the constant variable. 1 ml of 1%, 1.0% DCPIP is the constant variable. From the first paragraph, from the first instruction we already get. Fill the 5 ml syringe needle with the orange juice. So when I see the word orange juice, that tells me this is the manipulated variable. So this is the manipulated variable. Ensure that there are no air bubble traps in it. That means this is the precautionary measure. Because the air bubble will have oxygen. So if oxygen is present, they will affect the experiment result. So we have to make sure when we add this fruit juice, no air enters inside. Only the fruit juice enters into DCPIP. So the syringe should not have any air bubble. That's a precautionary measure. If you go further down, insert the tip of the syringe needle into the specimen tube and drip orange juice drop by drop into the DCPIP solution. Insert the tip of the syringe needle into the specimen tube. Drip the orange juice drop by drop into DCPIP solution. So dropping, maybe I remove this one. This is also precautionary measure. You must make sure the syringe is inside the DCPIP. The end of the syringe, the needle, should be inside the DCPIP solution. As you can see here, You can see it here that the needle is inside the DCPIP solution. You're supposed to add the fruit juice drop by drop. At the same time, you're supposed to stir it slowly until the blue color is decolorized. So in this paragraph or this sentence here, you have a few precautionary measures. Insert the tip of the syringe needle into the DCPIP solution. We add the fruit juice drop by drop. We stir the DCPIP solution slowly. All of that are precautionary measures that you need to follow when you're doing the experiment. If you don't follow these precautionary measures, most probably the experiment results will be affected. Then what do you do? What is the information you need to get after doing this experiment? You need to record the volume of you are supposed to record the volume of orange juice needed to decolorize the blue color of DCPIP solution. So the volume of orange juice needed to decolorize the blue color of DCPIP is the responding variable. You can get it from the instruction, responding variable. You repeat the experiment. Now you're going to use pineapple juice, and pineapple juice is going to become the manipulated variable. So this is manipulated variable, pineapple juice. So if you note all the information that you need, what is the manipulated variable, what is the responding variable, what is the precautionary measure, what is the constant variable, you can get it all from this instruction that is given. Many of this information that you get from this is needed to answer some of the questions later. So after that, you're supposed to record in the table that is given. So once you already get all the information you need from this instruction procedure, we can go to the next. You conduct the experiment, follow the instruction given. Based on the results of the experiment, you're going to fill in this table. So I have this is not that we don't know if this question coming out or not, but I've just randomly added some numbers here. Okay, so let's see what are the mistakes the student did. But before that, take note that the table is important, because from the table also you can get what is the responding variable. Sorry, it's English version. Volume of juice required to decolorize DCPIP solution is responding variable. The juice is the manipulated variable. So this is supposed to be manipulated variable. This is supposed to be the responding variable. You can also get it from the table. So take note of that, meaning later we need to identify all of these variables. There is many places where you can get the variable. You can get the variable from the first paragraph right on top here. You can get the variable from the procedure. You can also get the variable from the table. So just many places for us to get the answer. That is the beauty of paper 3. Almost all the answers are already in the question. So you just need to know where to find it. Now when you are filling in the table, one of the main things you need to take note is the decimal points must be same. So as a guide for us, they already have the value here 1.0. They could have just wrote here 1, but they didn't. They wrote here 1.0 to tell you that the answer we need is in 1 decimal point. So if here both I have, they given us 1.0, but I write here 1, so that's considered wrong. I'm not following the same decimal point. This must be 1.0. If you write 1, consider you won't get the 2 marks. They're going to penalize 1 mark because you are not following the same decimal point. That is 1. So both of these I have the same decimal point, but what I've done here, I take the average, I get 2.45. So now I have 2 decimal point. I'm not following what's given in the question, meaning again I'm going to get penalized. So this one should be 2.5 because the 5, if you have 5, that means 5, 6, 7, 8, 9, you need to add one more number. So 4 plus 1, you get 5. You have to bring it to the round number 2.5. Here also you can automatically see there is a mistake. All of this we need to change it to 0.0, we need to change this to 0.0, and we need to change this to 0.0. So make sure the decimal points are same. Important. Another thing is, in case you need to create the table on your own, when you create the table on your own, make sure the manipulated variable is on the left side. The top row is always the responding variable. In case you need to create the table, the right side here, the left side here must be manipulated. The top row must be responding variable. Make sure you have the heading of the table and make sure you have the unit. Without the unit also, they're going to minus 1. We don't know whether you need to create a table. If you need to create a table, make sure the unit is given. On this side, we didn't have. If there is a unit, also must give. Make sure the unit is there. Make sure the heading is there. Make sure you follow the same decimal point to get the 2 marks. That is the rule when it comes to table. They must have the same decimal point. After you fill in the table, the next question is, state one observation for the experiment. One observation. In the old syllabus, you need to provide 2 observations. Now you need only 1. So observation means writing back what you have in the table. You don't have to make any interpretation. You don't have to make any conclusion. Just write back what you see. Pemerhatian meaning just what you observe, you write back. So you have in this experiment, we have only 2 data. One data is for juice, orange juice. There's another data for pineapple juice. So we have only 2 juice. You can use either one. But let's say if you have 3 data, let's say there is one more, maybe let's say watermelon juice, for example. Watermelon juice. So if you have 1, 2, 3, if you have 3 data, preferably use, I think for the new syllabus it doesn't matter, but preferably use the highest or the lower. Let's say for watermelon juice I have 3.0. So when you compare these 3, this is the highest, whereas this is the lowest. Responding variable, this is the highest, this is lower. So if you are going to write the observation, you can write any 3, but preferably choose either the lowest or the highest. That will be the best. Like for example, if you have temperature affecting the enzyme, for example, enzyme action, temperature affecting enzyme action, we have 0 degrees Celsius, 25 degrees Celsius, 37 degrees Celsius, 45 degrees Celsius. The best observation is 37 degrees Celsius. That is the optimum temperature. So the 37 degrees Celsius will give you the shortest time. The reaction is fastest. So that is why we like the responding variable. When you are writing the observation, preferably choose the responding variable that gave us the highest or the lowest value, the extreme. If you have 3 data, like for this experiment there is only 2 data, so whichever you choose is the right answer. So make sure in the answer you have manipulated variable, responding variable, if possible constant variable, and the data. That is the technique for paper 3. Your answer must have all the variables that is needed in the experiment. So the answer I'm going to give is, it is the Malay version here. So for orange juice is the manipulated variable. Average volume of juice required to decolorize DCPIP solution is the responding variable. 3.3 ml is data. So what I can do is, when I look at this, I realize that I don't have the constant variable. If I go back, I see the constant variable is 1 ml. So I can make my answer better by adding in here 1 ml. So for orange juice, manipulated variable, average volume of juice required to decolorize 1 ml of DCPIP solution is 3.3 ml. This 1 ml is the constant variable. So every variable that we need is here. So they have to give you your mark. Manipulated is there, responding is there, constant is there, data of the experiment. Sorry, it's not 3.3, is, I forgot to change it. Orange juice is 2.5. That you get your 1 mark. just make sure you have all the variables. If any of the variable is missing, for example, the student wrote average volume of juice required to decolorize. This orange juice don't have, for example. So you might not get the 1 mark because your answer is not complete. So make sure, that is the trick. Make sure the variables are there. That you can get your full mark. From that, question 2. Inference based. Now is the time you need to explain to the examiner. You told the examiner that you are using 2.5 ml. The other one, pineapple juice, is 4.0 ml. So why there is a difference? Now you start to compare. When you write the observation, you just write what you see. You don't look at the others. When you write the inference, now you compare with the others. So this is 2.5, that is 4.0. So 2.5 is lower. When you say it's lower, you must explain why is it lower. You must give the explanation. That will be the inference. So when you write the inference, this is the inference I'm going to write. Average volume of fruit juice required to decolorize DCPIP. If you note, it is again corresponding variable. Average volume of fruit juice required to decolorize DCPIP is high. High is data. Actually, if it's 2.5 is low, not high, is low. It's low. And why is this low? We need to change the color. Okay, so the average volume of fruit juice required to decolorize DCPIP is low. You compare to 4.0 is low. Concentration of vitamin C in the fruit juice is high. Take note, the lower the volume of fruit juice, the higher the concentration of vitamin C. So average volume of fruit juice required to decolorize DCPIP is low. You compare with 4.0 is low. The concentration of vitamin C in the fruit juice is high. Take note, many students find this inference is hard. They normally tell us, I don't know what to write. The trick for this is, like for example, where did I get this concentration of vitamin C? There is normally two places you can get the answer for the inference. What to use in the inference? There is two places normally you can get. One is the first paragraph. But in this question, the first paragraph actually mentions contents of vitamin C. So you can write contents of vitamin C because it is there in the question. So you can use it. But do we have any other option? The other option is you find the question that will have calculation. So that is question D. Question D wants us to calculate, and what they want us to calculate is vitamin C concentration. So I see the word vitamin C concentration in question D. You are now actually doing question A2. Or when you're doing question A2, you look at the first paragraph, you're not that happy, contents of vitamin C. So you can actually go to the calculation question, and sometimes even from the table. The calculation question has the word vitamin C concentration, so we use that as the answer. So if I come back here, I make sure I follow what is in the question. So it's not concentration of vitamin C, it's vitamin C concentration. We follow what's in the question. So I wrote concentration of vitamin C. I'm changing it to vitamin C concentration. Why? Because the question has the word vitamin C concentration. I repeat, the question is in the answer is in the question. Paper 3 is like that. You just find the answer from the question, write it back. That is what I'm doing. I wrote concentration of vitamin C, but when I see at the back is the other way around, I change it. I want to make sure my answer follows what's in the question. So average volume of fruit juice required to decolorize DCPIP is low. Vitamin C concentration in the fruit juice is high. Here you just write what you see, observation. Here you explain what you see. Oh, compared to the pineapple, this one is low. Why is it low? Because it has more vitamin C. Example. You can also write, I chose here orange juice. You can also write pineapple juice. Your choice. Either one can. But if you have more than 2 data, choose the highest or the lowest. That will be the best observation. The highest or the lowest responding variable. That will be the best observation. For this, there's only 2. Either one is fine. So I repeat, to explain the inference, the keyword that you need, you get it either from the first paragraph or you get it from the calculation question. Most of the time you can get it there. So that is inference. After inference, we need to identify all the variables. I already told you where to get all the variables. You can get it from the first paragraph. You can get it from the procedure. You can also easily get it from the table. Table is one of the best place to get, easiest. The manipulated is here, the responding is here. And if you note, when it comes to manipulated variable, type of fruit juice, when it comes to responding variable, there is 2 answers. Volume of fruit juice required to decolorize DCPIP, and another one is vitamin C. Okay, what I have here, vitamin C concentration, follows. So vitamin, you can write either one, both of these correct. No need to write both. I will identify both. Why I always identify both? I know I need vitamin C concentration when I'm explaining the inference. For my inference, I need vitamin C concentration. So I will know there is 2 responding variable. Normally, I'll call this as the theory variable. You learn this in class, vitamin C concentration. What you did in the experiment, this one, I'll normally call it as the practical variable. Just for my understanding, the one we learn in class is the theory variable. The one you do in the actual experiment is the practical variable. You need to know both. You use this for inference, you use this for observation. This is considered another bonus 3 mark. This is actually the most important question. To make sure you can get marks for every other question, you need to know what is manipulated, what is responding, what is constant. Mainly responding and manipulated must know. Almost every answer will have manipulated and responding. So knowing what is it even before you answer the question is important. Like in the old syllabus, normally I'll tell the students, you answer question B first. Go straight to question B, identify all the variables. Make sure you know what is the variable even before you answer all the other questions. Why? Variable is the keyword. Make sure every answer you have the variable, you can get your marks. So this is question B. From question B down here, they ask for one hypothesis. The hypothesis is normally you use combination higher, 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 lower. Normally higher, 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 lower. The higher the temperature, the higher the rate of reaction. The higher the humidity, the lower the rate of transpiration. Normally is higher, 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 lower. That is the combination. Higher, 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 lower. I wrote it here, I think. So it's higher, higher or higher, lower. That is the normal method. But this higher, 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 lower, you can only use it if you have more than 2 data. You need to have minimum 3 data. Only then you can use this higher, 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 lower combination. The problem with this experiment is we have only 2 data. Orange juice, fruit juice. So because we only have orange juice, we have only 2 data. So we have to use the word compare. So if you note here, pineapple juice contains higher vitamin C concentration compared to orange juice. The word compare is, we normally use this compare if you cannot use higher lower combination. Even for experiment, like for example, you have the enzyme affected by temperature. Most of the time the experiment may be like this, maybe like this, or maybe like this. Okay. But when it comes to temperature, the graph actually goes up and then come down. So it's hard for us to use the word higher, 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 lower. So when we can't use that, we use compare. So rate of reaction. is highest at 37 degrees Celsius compared to this one, and this one. So anytime you can't use this combination, we use the word compare. So here we do the same thing. Pineapple juice contains higher concentration, vitamin C concentration compared to orange juice. All of these different colors is telling you the blue one is manipulated, the purple one is the responding, the red one is the data, and the red one is the word that you must use to answer the question. Higher is supposed to be the data. So you can use pineapple juice contains higher vitamin C, or you can write the other way around. Orange juice contains lower vitamin C concentration. Your choice. As long as manipulated responding is there. The hypothesis doesn't have to be the right one. For example, if I wrote here, pineapple juice contains lower vitamin C concentration compared to orange juice, you still accept it. When you do the experiment, you're going to prove it's wrong. Preferably write the correct hypothesis, which is pineapple juice contains, but again, I think this is a wrong hypothesis. From our experiment, which one has more vitamin C? Actually, orange juice has more vitamin C. So that means my hypothesis here, pineapple juice contains higher, is actually wrong. Wrong hypothesis. It's not, when I say wrong, I mean you will get the 2 marks. But we are not going to, when we do the experiment, we are going to prove this wrong. Preferably write the one you're going to prove. So better you write here lower. So lower vitamin C concentration compared to orange juice. This is a better answer. Not to say the earlier one is wrong, but the earlier one, when you do the experiment, you're going to come to conclusion the hypothesis not accepted. You didn't prove it. So preferably you write what you can prove in the experiment. So this one we can prove. Pineapple juice contains lower vitamin C concentration compared to orange juice, or orange juice contains higher vitamin C concentration compared to pineapple juice. Either one, 2 marks. Next question is calculation. If you are doing the calculation, as for me, I will make sure that the formula is already given, but there is a mistake in the formula, which is they are asking for concentration. They already give us the value, milligram, milliliter, -1. But in here they wrote 1%. So you times with 1% if you want percentage of vitamin C. So the heading here and the calculation is wrong. So you times with 1% if you want to get percentage of vitamin C. Here we want concentration of vitamin C. So this is wrong. You are supposed to actually multiply this. So ini salah. You're supposed to multiply this with 1 milligram milliliter -1. Okay, you multiply with 1 milligram milliliter -1. Don't worry, the actual SPM question, they won't be mistake. Generally, very rarely it happens. Generally we can say no mistake. This one I think from one of the, either maybe from school, maybe they had made unknowingly they made a mistake. Now man, so we just make sure it's one. Check whether it's concentration or percentage. Percentage you times 1%. Concentration you times 1 milligram milliliter -1. So when I'm doing the calculation, I make sure I lead the examiner to the answer. So I make sure I write everything. Vitamin C concentration in orange juice. There are students who just write orange juice and then they do the calculation. Write the full sentence. Vitamin C concentration in orange juice. Write the full sentence. Don't give chance for the examiner to penalize you. You write the whole sentence. They have to give you the marks. Same thing with here. I make sure I write unit in all the calculation, including the answer. So the calculation and the answer, I make sure I have the unit. Again, I want to make sure they can't penalize me because everything is given. So you do the calculation. Actually, when I started to do the calculation, I got 0.4. I wrote 0.4 milligram. This 0 I didn't write. When I do the second calculation, I got 0.25 because I get 0.25 here. Here I come back and I change it to 0.40 to make sure I have the same decimal point. If you have very long number 0.3333 for example, normally for SPM level 2 decimal point enough. So bring it to 2 decimal point. Like here the answer I got is 2 decimal point, but here I had only 1 decimal point. I make it 2 to make sure the value is same, the decimal point is same. Practical, all of this is important because you're going to compare. When we compare, we must use the same decimal point. So that is calculation. Just follow the formula given, make sure you have all the units, and make sure you write the full sentence. You can get your 2 marks. After that, you need to write operational definition. So this is for many students this is hard to answer. What is the operational definition? Many students, they write, because they see the word definition, they write what they learn in class. That's what many students do. What is transpiration? Transpiration, let's say the question, the experiment is something to do with transpiration. For example, what is transpiration? Transpiration is evaporation of water vapor from the surface of plants. They write back what they have in the textbook. That is theory. The question didn't ask for the definition of transpiration. The question asked what is the operational definition. The word operational means in this experiment, what is transpiration? Like in this question, in this experiment, what is vitamin C concentration? This question is confusing us. Now earlier they had contents of vitamin C, then they had vitamin C concentration. Now they have concentration of vitamin C. That means you can use this as the answer. I took the trouble to change it earlier, but don't worry, all this kind of confusion doesn't happen in the actual exam. Actual exam, it will be, if one place they use vitamin C concentration, all the place they use vitamin C concentration. Like I think maybe the no comments. Okay, so anyway, so here operational definition means in this experiment, what is concentration of vitamin C? So we are going to use a technique to answer this operational. End of the day, manipulated variable, responding variable, constant variable. End of the day, make sure you have that in the answer. So what we are going to do is this. The technique to answer is question is responding variable plus constant variable. When I say question means what is asked in the question, this one. So concentration of vitamin C is responding variable and constant variable. Meaning in the first sentence, you need to have responding variable, you need to have constant variable. What is missing? Manipulated variable. So manipulated variable, you write it in the second sentence. The question is affected, must have. The question is concentration of vitamin C. Concentration of vitamin C is affected by manipulated variable. Use this technique. Meaning you must have all the variables in the answer. That's basically it. So if you're using this technique, what will be the answer for this question? Vitamin C concentration, that is the question, is volume of fruit juice required to decolorize 1 ml of DCPIP. Maybe I change this because this is the question, so I change it into black color. So what's in purple is responding variable. What's in red is manipulated variable. I even have constant variable, which is the fruit juice. I repeat, purple color responding variable, blue color manipulated variable, red color constant variable. Every variable is there. Also take note, when we write operational definition, we don't have to look up at the ceiling to get the answer. It's in the question. The answer is already here. This is the 3 variables you identify earlier. Make sure you have all of this in the answer. That is operational definition. The main answer for operational definition is this. Responding variable must be in the answer. So if you go back to this, Vitamin C concentration is volume of fruit juice required to decolorize 1 ml of DCPIP. I need the second sentence. The second sentence is this. Vitamin C concentration. I change this because that's the question. So vitamin C concentration is affected by the type of fruit juice. Type of fruit juice is manipulated variable. That's how easy inference, operational definition supposed to be. The technique is responding, constant, manipulated. Make sure you have in the answer, and that's basically it. So this will be my answer, but also read the question carefully. Like this question wants us to write a concentration of vitamin C. Because they ask concentration of vitamin C, because they ask for concentration of vitamin C, we write this answer. If let's say the same question was this, operational definition of vitamin C. If they ask you operational definition of vitamin C, what you need to do is you need to remove this and you need to remove this. That is quantitative. So the quantitative words, anything to do with calculation, we remove. Vitamin C is fruit juice required to decolorize 1 ml of DCPA. Because they didn't ask for the concentration. If they ask for concentration, then you mention the volume. If they didn't ask concentration, no need volume. So check the question properly whether they want vitamin C or whether they want concentration of vitamin C. Make sure you read the question carefully when you're answering. So this is operational definition. Hopefully this technique makes your life easy in terms of how to answer. Every answer comes from here. Normally, I think I already mentioned, normally in the old syllabus, I tell the students, come straight to question B. Come to question B, identify all the variables before you answer any other question. Because every other question needs the variable. You have the variable, you get your mark. So the last question. The last question is prediction. So this is, the last question is prediction. So normally what happens is, the prediction is, we change the constant variable. That is what normally happens. We change the constant variable. Throughout the experiment, constant variable was constant. So what we are going to do now, the last question we change it and we ask you what is the effect. We ask you to predict. In this experiment is 1 ml DCPIP. So we are not changing that. In this question, they are changing the manipulated variable. Earlier we used fresh fruit juice. Now they are leaving the juice for 4 hours. So it's no more fresh. It's not a fresh, and you naturally know if the fruit juice is not fresh, they are going to have less vitamin C. So they are asking you to predict. So normally students will write the fruit juice has less vitamin C. Actually, the marking scheme has that answer. It has less vitamin C. But I'm going to go with what we learned in the previous syllabus, which is in the last question, when you're making prediction, you must use the data from the experiment. So the data is important. The marking scheme for this question didn't have the data. I'm putting in the data here because when we make the prediction, we need to use what we already know from the experiment. What we know from the experiment is this. This is a calculation we made. We know orange juice is supposed to have 0.40 milligram, whereas pineapple juice 0.25. So this is the data from the experiment. A fresh pineapple juice is supposed to have 0.25 milligram milliliter -1. If it's not fresh, the value should be lower than 0.25. That is what we mean by using the data from the experiment. Instead of just telling they have lower concentration of vitamin C, I'm going to use the data and I'm going to write this answer. Vitamin C concentration in the pineapple juice is lower than 0.25. This is the data from the experiment. But actually the marking scheme for this paper, they just use this one. They didn't have this last data. But I'm going to ask you to write the data. It's lower than 0.25 milligram milliliter negative 1. The question also asks you to explain your answer. So the explanation is this: the fruit juice is not fresh. Vitamin C in the fruit juice is oxidized when exposed to air. The prediction, try to use the data from the experiment. You already do the calculation earlier, use it to support your answer. A student who is telling the fruit juice concentration of vitamin C is lower, and a student who tells is lower than 0.25 will get it's a more precise answer. So that's basically it. That is how you answer a paper 3 question. Every single answer, make sure you have manipulated variable, responding variable, if possible constant variable. The whole experiment is about that, variable. You can have the variables in the answer, you can get your full marks. That is what I did. That is why for me this is the most important question. Identifying the variables. If I know the variables, generally I can get the full marks. Another question that many students are concerned is, what if it's an unguided question? If it's unguided question, you need to get the manipulated, you need to explain to the examiner how you conduct the experiment. They give you 4 marks. Same thing, in that when you're explaining to the examiner wants to know how you control manipulated, how you get responding, how you get control. That's the 3, plus precautionary measure. So when you are writing procedure, when you're writing the procedure, when you're giving instruction, there are certain keywords we need to use. You can get it from here. So this is the procedure that is in this question. From here, let's say it becomes unguided question. You need to inform the examiner how to control the variables. There are some keywords we need to use. So this is the 3 that you need. You need manipulated variable, responding variable, constant variable. Precautionary measure is already there, you can read. So for manipulated variable, we need to use the word use. So the word use must be there. Use fresh juice and fresh pineapple juice as the manipulated variable. Or you can say use different type of fruit juice. Use a different type, because fruit juice is manipulated type of fruit juice. So use different type of fruit juice. I normally like to mention what I'm using. So use different temperature of water bath. 0 degrees Celsius, 25 degrees Celsius, 37 degrees Celsius, 45 degrees Celsius. I like to mention what I'm using. So use fresh orange juice and fresh pineapple juice as manipulated variable. You can just write use different types of juice. Okay, correct. But as I mentioned, I prefer to write what I'm using. Responding variable, you must have the word measure and record. Measure, check, there are times, for example, photosynthesis experiment, you count the number of bubbles that is coming out from the plant, released by the plant. So you can't write there measure the number of bubbles, count the number of bubbles. Like if it's distant, you can use measure. So check what you're supposed to do in the experiment. So use the correct word, measure and record, count and record, for example. So measure and record volume of fruit juice required to decolorize 1 ml of DCPAP. That one you already know, we saw in the question. But the keyword, measure and record. Another important thing for responding variable is you need to mention what is the device you use to get your responding variable. In this experiment, to get the volume of fruit juice, we use 5 ml syringe. If let's say you need to measure temperature, measure and record temperature of the experiment by using thermometer. No thermometer, no marks. Measure and record the length of the gas bubble by using a meter ruler. No meter ruler, no mark. Most of the time, the apparatus the device will be there. Like the photosynthesis question earlier, you count the number of bubbles, you use your eyes. No need to mention your eyes. So rarely it happens. Almost all the experiment, you have a device to measure and record responding. The device, make sure you mention. As I was telling earlier, you are lucky because the device, the apparatus is in front of you. So you generally know, you can predict more or less which one you need to use. Previously, they only have the question paper, so they have to, they have to remember what they read from the textbook. You have an advantage; it's in front of you. So from there, you can get what is the device. Constant variable. The keyword is you must use the word fix. Sometimes they use the word maintain. Even set also can, but fix is the most commonly used. Fix the volume of DCPIP at 1 ml for every experiment. Set the temperature at 37 degrees Celsius for every experiment. So fix or set can. Check the question or choose the right one. Fix this. The word that I underline is all important. Must be in the explanation. This is if it's unguided. You need to, the first question, you need to explain to the examiner how you are going to do the experiment. These are the points you need. The precautionary measure, you can use any one of these that we underline in red. You can use any one of them. That is the precautionary measure. Other than that, the procedure, if you need to write, use these keywords, you should be able to get the marks. So this is paper 3. So by right, as I mentioned earlier, I'll mention again. As for me, paper 3 is supposed to be a bonus question. You might be concerned in the sense, you don't know what experiment is going to come out. You read your textbook. The textbook has all the experiments. Just go through it. Take note for every experiment the variables. Take note of the variables. And there are some experiments that are more famous for exam, like vitamin C, yes, enzyme experiments, whisking tube experiment, food test experiment. There are some experiments that are more important in exam. So we also need to consider experiments that can be easily done within the 40 minutes. There are some experiments we need longer duration. So we also have to consider, is this experiment can be done within the 1 hour, 2 hour time of the experiment. So hard to predict, but if you have got the techniques is important. You know the techniques, you can get all the answer from the question. So if you know the technique, this is an easy paper. As for bio, physics, chemistry, no comments, not my department. Bio, if you follow the instructions, should be able to do well in this experiment. So I'm discussing DCPIP because the rumors going around DCPIP is going to come up. So I've discussed this. I have my own Soalan Ramalan, not Soalan Bocor, Soalan Ramalan, what I think could be the question. So I'll do another video. So the next video is my predicted question. This question is for you to know the techniques. The next video is what I think could be the exam question. I'll see you in the next video.